Good morning, everyone. Today we will be discussing a very important group of bacteria in medical microbiology, the non-fermented gram-negative bacilli. We will focus mainly on one of the most clinically significant species, Pseudomonas aeruginosa. This bacterium is a major cause of healthcare-associated infections, and understanding its characteristics is essential for your future clinical practice. Let's begin with a general introduction. Non-fermenting gram-negative bacilli are a group of bacteria that do not ferment sugars. They are strict aerobes, meaning they require oxygen to grow. This group includes several genera such as Pseudomonas, Acinetobacter, and Stenotrophomonas. These bacteria are found widely in the environment and are usually harmless to healthy individuals. However, in certain situations, such as hospitalized or immunocompromised patients, they can cause serious opportunistic infections. These are often referred to as healthcare associated infections or nosocomial infections. Now let's focus on Pseudomonas aeruginosa. First, its habitat. The bacterium is found everywhere in the environment, especially in water. You can find it in sinks, taps, siphons, and even in disinfectant solutions. One of its key features is its ability to form biofilms, which are communities of bacteria that stick to surfaces and are protected from antibiotics and the immune system. Under the microscope, Pseudomonas aeruginosa appears as long, slender gram-negative rod with rounded ends. It is motile thanks to a single polar flagellum which looks like a tail. This mobility helps the bacterium move toward nutrients or away from harmful substances. Grows easily on standard culture media. You don't need special conditions to culture it, which is why it's often found in hospital environments. It is a strict aerobe, so it requires oxygen to grow. A key laboratory feature is that it is oxidase positive, which helps distinguish it from Enterobacterales. Unlike many other gram-negative bacteria, Pseudomonas do not ferment sugars. That's why we call it non-fermentative. We can identify Pseudomonas aeruginosa using several methods: biochemical tests, Malditov mass spectrometry, or molecular biology. A very distinctive feature is the production of two pigments, piocyanin, which gives a blue-green color, and pioverdin, which is yellow-green. These pigments are not only useful for identification, but also play a role in the bacterium's virulence. Now, an important clinical point. Pseudomonas aeruginosa is naturally resistant to many antibiotics. This is called natural multi-resistance. For example, it's resistant to several beta-lactams and some aminoglycosides. This limits our therapeutic options. However, some antibiotics remain active. Among beta-lactams, we have ticarcillin, piperacillin, ceftazidime, and imipenem. Aminoglycosides like tobramycin and amikacin and fluoroquinolones like ciprofloxacins are also effective. But be careful. Acquired resistance can emerge quickly during treatment, especially in chronically infected or hospitalized patients. Now, let's move to pathogenicity. Pseudomonas has several tools to cause infection. First, it adheres to host cells using pili, flagella, and fimbriae. Then it invades tissues using enzymes like protease, elastase, and phospholipases, as well as exotoxin, which inhibits protein synthesis in host cells. The pigments we mentioned earlier also contribute to tissue damage. To spread and survive, it produces leukocidin, which kills neutrophils, and its lipopolysaccharide or LPS acts as an endotoxin, triggering severe inflammatory responses like septic shock. Some strains also produce capsule called alginate, especially in patients with cystic fibrosis. This capsule protects the bacterium from the immune system and antibiotics. Finally, it forms biofilms on medical devices like catheter, making infections very hard to treat. A biofilm is a structured community of microbial cells that is attached to a surface and enclosed in self-produced extracellular polymeric substance, also known as slime. This matrix protects the microorganism from environmental stress, including antibiotics, disinfectants, and the host immune system. Biofilms can form on both living tissues and inert surfaces. They are a major cause of persistent and chronic infections, as well as healthcare-associated infections. This image shows how biofilms form on a catheter. These bacterial communities are highly resistant to treatment and are a major source of persistent infections. As I mentioned earlier, Pseudomonas is an opportunistic pathogen. It mainly affects patients who are hospitalized for long periods, especially those in intensive care, hematology, or urology. Other at-risk patients include those with cystic fibrosis, bronchiectasis, diabetes, burns, or urinary catheters, and those who have received multiple antibiotics. Mortality can be as high as 50% in severe infections. Clinically, Pseudomonas can cause a wide range of infections: urinary tract infections, pneumonia, skin and soft tissue infections, especially diabetic foot ulcers, sepsis, bone and joint infections, and even endocarditis. Pseudomonas aeruginosa is the fourth most common cause of nosocomial infections, accounting for about 10% of all hospital-acquired infections. It spreads from patient to patient or through contact with contaminated environmental sources. These include disinfectants, respiratory equipment, catheters, food, and even towels. This is why hygiene is so critical in hospitals. Finally, let's talk about treatment and prevention. Antibiotic therapy must be guided by the natural resistance profile and, ideally, by an antibiogram. Active antibiotics include ceftazidime, imipenem, amikacin, and ciprofloxacin. Prevention is just as important. Strict application of hygiene rules, especially hand hygiene, and limiting the use of medical devices like catheters to only when absolutely needed. To summarize, Pseudomonas aeruginosa is a classic example of an opportunistic pathogen. Its environmental reservoir, natural resistance, and the ability to cause severe infections make it a major challenge in healthcare settings. Understanding its biology and epidemiology is essential for preventing and treating infections effectively.